Hello, my name is Kumar Shalubhai. I'm CEO and CSO of Tiziana Life Sciences. Tiziana Life Sciences is a dual listed company. It is listed in AIMS, London, as well as NASDAQ. And the symbol for AIMS is TILS, and for NASDAQ, it is TLSA. What you will hear today in my presentation, that our company is a clinical stage company. We have 4 assets which are already in clinical studies, and we have the clinical data which makes me believe that our programs are de-risked. And in addition to solid diversified pipeline, we also have people who have done bench to market in the past. So let me, before I go into details of the presentation, let me just show you the forward-looking statement. Of course, you don't have to read it, but I just want you to be aware of before considering investment in our company. Let me take you to slide number 3. Quickly, what I would like to emphasize here, that I'm a biochemist. I've been in pharmaceutical industry for 30 plus years. I've had a lot of success from bringing compounds from bench to market. I was also I was co-founder of Synergy Pharmaceutical, a company that was founded on my pioneering technology on a new concept for treatment of gastrointestinal diseases. And that technology has now led to approval of my first drug called Trulance, Trulanc. It is doing very well. And my second drug, Dolkenatide, has already completed phase 2 trial. So we've done it before, and we have the experience from bench to market. In addition to that, I have Professor Napoleon Ferrara, who was the discoverer of Avastin, a multi-billion-dollar drug from Genentech. He's been very helpful in development of our antibody assets. Then I have Howard Weiner. He's a professor at Harvard Medical School and top 5 people in the world to do so. And we're very excited to have him. And my co-founder and my friend for many years, Gabriel Charon, has been also very successful in bringing a drug from while he was in SIGA technology. So within this small Tiziana Life Sciences company, we have 3 people who have done it before, from bringing a compound from bench to market. And that experience is very valuable. And that is what, in a way, is the most exciting for my team. This slide gives you investment highlights of the company. And as you will see, that Tiziana Life Sciences is doing something totally transformational technologies, in the sense that, as you know, currently antibodies are given intravenously. And that is a complex affair and very costly. More importantly, the patient compliance is very poor, because when patients receive infusion of antibodies every 2 weeks for 2 hours, the blood level in the first 2 hours is very high. And that is what is causing a lot of toxicities. Our approach is to give antibody where action is needed. For example, oral administration is good for diseases that are in GI tract. So we completed phase 1 trial with oral administration and showed beautiful data and the phase 2 trials will be starting soon for Crohn disease. For nasal trial, again, a totally innovative approach. The antibody is delivered through nostrils, and the delivered antibody then activates mucosal immunity to provide anti-inflammatory effect in brain. Very important aspect of our technology is that when antibody is given nasally, it activates mucosal immunity in the brain, which results in upregulation of Tregs, and Tregs have the ability to cross through blood-brain barrier to provide anti-inflammatory effect wherever in brain the inflammation is. This is exciting development for the reason that in neurodegenerative diseases, many drugs have failed because they are not able to cross through blood-brain barrier. My third program is on COVID-19 treatment. And again, the approach is very logical and simple. Concept behind that is that COVID-19 infection occurs in lungs, and that is where virus grows, produces inflammation in lungs, and that inflammation is because of a cytokine called interleukin 6 or IL-6. Currently, the antibodies like Roche antibody, Actemra, or Capzara of Sanofi Regeneron is delivered through intravenous or subcut in some cases. But the drug delivered through these routes reaches lung in very small amount. Therefore, you have to probably give much higher dose. Instead, our technologies, and we are the first one to deliver interleukin IL-6 antibody directly through inhalation route to lungs where the action is needed. And I think this is a very innovative approach. We have filed patent, and I think we were the first one to show that, yes, this can be done, and this could have much better effect. Now, on, one other program that I have, and again, you will see that that is very innovative treatment for treatment of hepatocellular carcinoma. We completed phase 2 trial, and in phase 2 trial, we showed that our drug is outperforming the other existing drugs. So therefore, just to summarize the investment highlights of Tiziana Life Sciences, we have 4 programs. All 4 of them are in clinical studies. And so far, clinical data that we have already achieved, accomplished, makes me believe that our programs are de-risked. And this is something very exciting. And let me take you to slide number 6. It is exactly what I've already told you. But what you see here is that so far, immunotherapies are done through intravenous administration. And we have overcome the problems of intravenous administration with alternative routes of oral, nasal, and inhalation. And this approach is, in a way, a logical approach, which provides local action of antibodies wherever you have inflammation in body. So let's go on to slide number 9. This is a very important slide. And what I want to emphasize, on your left hand, you see an antibody shown in red color. It is called OKT3. It's a murine or mouse anti-CD3 antibody. This antibody was developed by Johnson & Johnson. It was approved by FDA, and this drug was taken off the market pretty soon because this antibody, being a mouse antibody, produced severe immune reaction. And therefore, it was taken off the market. But clinical data were so compelling that other companies like GSK and Nuvion, they tried to humanize this antibody. Humanization of antibody is that they make some mutations within the antibody and make it look like human antibody so that it can evade human immune system. All of these companies were successful in immunization of the antibody. Unfortunately, long-term use of these antibodies still produce immune reactions. And that is where these other antibodies didn't go anywhere. I'm very pleased to tell you that my antibody is the only antibody which is fully human. And we have shown in clinical studies that my antibody does not produce any immune reaction. Therefore, Foralumab, which is my antibody, is identical to OKT3, except that my antibody does not have the issue of immune reaction that OKT3 had. And therefore, this becomes a unique asset, and its opportunities or potentials are in a number of areas. However, being a small company, I'm very, very focused on Foralumab for oral administration in Crohn disease. We have completed phase 1 study. And now we are going to start phase 2 study in Crohn disease pretty soon. We have completed nasal trial, which is being developed for progressive MS. And the data that we publicly reported is with oral and nasal clearly demonstrate that when antibody is given through oral or nasal, it does not have the toxicities which are commonly observed with anti-CD3. And that's very exciting. More importantly, we were the first one to show that when antibody is given, for example, let's say nasally, it upregulates Tregs or T regulatory cells. T regulatory cells are, in a way, our body defenders, and they are completely depleted during inflammation and oncology. So our approach is to give antibody to upregulate or to strengthen our own body defense, and then let body defense fight with the disease. And I think that is a very physiological approach, very close to natural way of treating these drugs. So let's move on to slide number 10. And what I quickly want to emphasize here is that we filed a patent on oral administration and nasal administration in 2016. This patent was granted last week. Now, this patent is the first ever patent in this area. So my technology, which is to successfully lyophilize the antibody and then encapsulate in enteric-coated capsule, is the first one. And this antibody is applicable to all anti-CD3 antibodies. In addition, our antibodies, our patent estate also covers other antibodies as well. So I think this becomes a very important growth driver for Tiziana Life Sciences, and potential for our technology with some of the other very well-known FDA-approved antibodies is pretty good. So let me just quickly show you on slide number 11. I'm not going to go in details of it because all I want to show you is that we successfully completed nasal the trial with nasal administration, and data were very, very good. We are very excited to move on to phase 2 trial. Next slide is 12. There again, it is for oral administration of Foralumab. Again, the same data we observed, which is very promising, that when antibody is given through oral or nasal, it does not produce the toxicities that are so common with intravenous administration of anti-CD3. So in a way, what I'm trying to emphasize here is that these alternative routes of administration seems to be working, seems to be minimizing toxicities, and this could really transform the way immunotherapies with monoclonal antibodies are done currently. The bigger question is, yes, you can give antibody through these alternative routes, but does it work? And let me show you a study that was done by Professor Scott Snapper. Scott Snapper is a professor at Harvard Medical School, and he is the name in gastrointestinal diseases. So let me take you to slide number 13. This slide shows you the clinical data obtained by Scott Snapper. We did not fund this trial. We did not pay for it, but it was Scott's interest to see if this alternative innovative therapy really works. And what he reported is amazing. It's music to my ears. And this is already published. What he showed, that when anti-CD3 is given orally, 3 out of 6 patients showed clinical response. 1 patient showed complete remission of the disease. And I can tell you, I have not seen that kind of clinical response, even with some of the existing drugs for GI treatment. So this is very exciting news for us. Let me also show you another study that was also done by a third party, and we did not do it. And let's move on to slide number 14. So this study was done by Howard Weiner. He's professor at Harvard Medical School. He's also the director of N. Romney Center. at Harvard University. He is the top 5 people in the world. He's very famous, brilliant scientist, and very humble person. He was also very excited with our new approach. And so he did the trial in 36 patients of NASH, 9 patients per group, dose ranging trial from 0.2 to 5 milligram. And what he showed that when you do the antibody administration through oral once a day for 1 month, the antibody treatment is completely safe. It did not have the toxicity that are so commonly observed with anti-CD3 when given intravenously or subcute. More importantly, he also looked at the efficacy biomarker just to assess if antibody when given orally works or not. And what he showed was that the efficacy biomarkers for insulin dependency is reduced by 50%, along with reduction in liver enzymes. And these are the clinical endpoints that FDA looks at for approval of drug. So it shows that yes, antibody can be given orally and it works. But most importantly for Howard Weiner, he also looked at the upregulation of T regulatory cells in blood, and he showed that when antibody is given orally, it upregulates Tregs in blood. So in a way, think about it this, the Tregs are our body defenders. They circulate in blood to provide anti-inflammatory effect wherever we have inflammation, which is a physiological process. And what we are trying to do is upregulate Tregs or strengthen our body defense and let the body defense fight with the disease. This phenomenon of oral administration existed with the life of humankind from the very beginning. Now, this phenomenon, the inspiration for this approach comes from a very natural phenomenon. That is that when a child is born, child gets immunity through mother's milk. And that's exactly what we are doing. In addition, there are ample examples of oral vaccines which are so successful. So that makes me believe that, yes, we are on the right approach. And I'm very comfortable moving forward with these alternative approaches for treatment of human diseases with monoclonal antibodies. If we succeed in any one of these, that would change or that would revolutionize the way immunotherapies is done. And as you know, immunotherapy is a $100 billion market currently, and it is growing very rapidly. So that is something very excited. We are very excited about. Let me move on to another one, which personally as a scientist, I am very excited and I want to expedite that. That is for treatment of COVID-19. It's urgent. We need to have it now. And our approach is that use this anti-IL-6 antibody, receptor antibody, which depletes circulating level of IL-6. And as you know, or as you might know through the literature or on TV, that the cytokine interleukin 6 or IL-6 is the mother cause of cytokine storm, which produces hyperinflammation in lungs, leading to respiratory failure and death. So there have been successful data from Actemra of Roche, which is also anti-IL-6R antibody. Ours is also the same, but our antibody has distinct advantages. Number one, it's a human antibody. Number two, it depletes IL-6. And number three, that we are delivering the antibody directly in lung where IL-6 is in excess. Remove or deplete IL-6 from lung should provide immediate relief to these COVID-19 patients. And that is something that I have already patented. My patent not only covers my antibody, but it also covers other antiviral agents or drugs. Say, for example, remdesivir of Gilead is also covered in my patent. So that gives us that gives a lot of potential to our company, our technology, that it could hopefully be successful in the future. Now, this is all about the antibody. Now, let me quickly spend a couple of minutes on treatment or our approach for hepatocellular carcinoma. Now, hepatocellular carcinoma, as you know, is a complex cancer. It is caused by hepatitis B virus, hepatitis C virus, aflatoxin, alcohol, and a number of other factors. So within a given population of these patients, there exists a complex heterogeneity. Complex heterogeneity in the sense that there are multiple mechanisms causing this tumor to grow. Now, if that is the case, and if your drug is specific for one mechanism, then you may get clinical response, but only in a subset of patients. And that's exactly what is happening. Sorafenib is a standard of care. It's an inhibitor of tyrosine kinase pathway. And the response rate is just 13%. So I began asking these questions. What happens to the 87% of the patients who are not responding? And I believe that is where is the unmet medical need. So my approach is to take a compound that would inhibit multiple mechanisms which are underlying this cancer. And that should provide a better clinical outcome. So let's go on to slide number 18. And Milciclib is a broad spectrum inhibitor of cyclin-dependent kinases. And we did, this compound was being developed by Nerviano Medical Sciences in Italy. And they did 2 phase 2 trials successfully. Successfully in the sense that Milciclib treatment in these 2 cancers in phase 2 trial with 104 patients showed that Milciclib met primary as well as secondary endpoints. We in-licensed this compound and did a trial with hepatocellular carcinoma, which is a deadly cancer. Now, when we were doing this trial, FDA insisted that we have to get sorafenib resistant patients, in the sense that, which is ethical also, that sorafenib is a standard of care, and these patients are running out of time. So therefore, they should be given some treatment. So we enrolled sorafenib resistant patient. These patients' prognosis is very poor. They live anywhere from 2 months to 5 months. When we designed the trial, we kept the trial duration for 6 months, 1 more month than these patients live maximum. And I'm pleased to tell you that 14 out of 28 patients, that is already 50%, completed the trial. No death. Treatment was well tolerated. No death is very uncommon in this group of patients, and we did not see any. But more importantly, 9 patients out of these 14 patients requested for compassionate use to continue treatment with Milciclib. Compassionate use is when patients feel happy about the drug, they like the drug, they request, and we are obligated to provide the drug. I'm excited to tell you that 9 of these patients, 7 of them, have completed anywhere from 9 to 16 months. And 2 patients are still continuing with the treatment, and they are in their 19th month. Compare 19 months to 5 months of maximum survival. Also, the other clinical endpoints that FDA looks at it, which are given on this slide at the bottom in bold, Milciclib seems to be outperforming existing therapy for hepatocellular carcinoma. This is very exciting development, and I believe Milciclib has potential to be developed for global market. Now let's go on to slide number 20. I just want to show you that we presented 4 posters in ASCO. And ASCO is American Society of Clinical Oncology. 2 of our posters were given special recognition. And what is so important here is that we developed a test which is for prognosis or for prediction of metastases in postmenopausal women for breast cancer. This is a huge market, and currently the market leader is Oncotype DX, which is marketed by Exact Sciences. Exact Sciences acquired this technology from Genome Health for $2.8 billion. We did the trial with 3,200 patients, side by side, comparison with Oncotype DX, and we showed that our test is 40% more accurate and it is superior to Oncotype DX. Now, this technology is hidden in Tiziana Life Sciences because Tiziana Life Sciences is a therapeutic company. Therefore, my board allowed me to spin out this technology as a separate new company. And that should happen in a couple of months. And that is beneficial for shareholders of Tiziana Life Sciences as well. They will also get a piece of the new company. So I think that that is exciting for both Tiziana Life Sciences as well as the existing shareholders of Tiziana Life Sciences. Okay, so let's move on to slide number 22. This slide just shows you our intellectual property. We have 256 patients, and we are very comfortable with our solid intellectual property, not only covering composition of matter, but all manufacturing technology and all other things. And it takes us all the way up to 2043 for Foralumab, for Milciclib, and for TZLS 501. And we are expanding our IP portfolio by filing new patent applications. Let me quickly also take you to slide number 23. These are the major catalysts that are upcoming. Second quarter of 2001, we'll be reporting phase 2 data, oral administration in Crohn disease, which is a huge market. We would also report phase 2 data in nasal administration for progressive MS, which is unmet medical need. We will also start clinical study in COVID-19 in first quarter of 2021, which is very important and urgent need. And then we will also initiate the global trial with Milciclib, hopefully soon. All right. So thank you very much for your time, and I would be happy to take your questions now. Thank you.
We are joined today by Tiziana's founder and executive chairman, Gabriel Cerrone, and CEO, CSO, Kunwar Shalawai. Welcome, gentlemen. We have a lot of questions already, so we'll just jump right into things. The current ATM is due to expire tomorrow. Will this be extended, or has the funding been secured?
I would let Gabe answer this question. We could only answer information that's been publicly disclosed.
Okay. Very good.
We can't answer non-public material information on this call.
Sure. Okay. Very good. Our next question, given the pipeline size and depth, together with the current cash burn, how long does the company expect to run before more funding is required?
Thank you very much for joining the webinar. And, I'll try to answer this question. So, as you know, and it's publicly known that we raised money about 3-4 months ago for $10 million. And so we have enough cash on hand to take us through all the way up to 2021. And by that time, we would be reporting significant major catalysts. For example, we will be reporting in second quarter of 2021 nasal administration with Foralumab in multiple sclerosis. We would also report the initiation of a study in COVID-19, and we will also report in third quarter of 2021 the oral administration of Foralumab in Crohn disease. So these are the three major catalyst points, and we would be able to accomplish all of this through the cash we have on hand. So we don't need to necessarily go for any fundraise in the near term. Besides, in the near term catalyst within this year, there would be also some updates on our expedited clinical development plan for COVID-19, and we will report those shortly. And then we will also provide update on some of the corporate events and business development activities. So we are very comfortable with the cash on hand right now.
Very good. Thank you, Shalu. And our next question, somewhat related, will additional funding be secured by way of a deal with large pharma, such as upfront payments, for example?
As we have already publicly announced that there has been a lot of excitement, and we've been approached by several big pharma, and we also have confidentiality agreement with some of them. So it is always good to join hands with big pharma, as that is very helpful to expedite clinical development and also explore other opportunities. So but currently our singular focus is to accomplish major catalysts and explore business development opportunities with other big pharma. And so we've been in touch with, and as soon as we have updates, we will put it out in public.
Very good, thank you. And our next question, can the stem printer approval be accelerated?
Yeah, so our plan with stem printer is to launch it in Italy first, because it was developed in Milan at the European Institute of Oncology, which is the most prestigious center for treating breast cancer patients. So our pathway is to file a CE mark in Europe, which allows us to launch the product as early as 2021. So our plan is to launch it where we know there's already a local market. As we've mentioned, it's been in 3,200 human patient samples already. So there's been a robust supply of samples from the European Institute of Oncology. And so the shortest path to getting to the market is to launch it via a CE mark in 2021 in Europe. And then from there, we plan to expand to the UK, the rest of Europe, and then to the United States.
Very good, thank you. And another question on stem printer. How will stem printer be marketed? Will an out-licensing or commercial partner be sought?
Well, we are evaluating initially in Italy. We are going to launch it internally ourselves, and as we expand into other countries that are not home markets, we plan to look at opportunistic deals that come to us. So it'll be evaluated at the time, depending on how interesting the proposals are.
Okay, thank you. And our next question, what are the implications for Tiziana's prospective COVID treatment and yesterday's Roche results from their trial of Actemra IL-6 that failed to meet the endpoints?
I'll take that question. So first of all, this is an excellent question, and I must thank the person who has asked this question. And this also gives me an opportunity to tell you more about the distinctive approach we are following for COVID-19 treatment. As you all know, this virus grows in lungs and causes inflammation within lungs, producing cytokine storms that leads to respiratory failure. So Actemra in this study was administered by intravenous, and as you would expect, a small amount of this antibody reaches the lungs. By contrast, our approach is to directly deliver anti-IL-6 antibody in lungs. Another distinctive feature of our antibody is that our antibody is the only antibody that also depletes circulating level of IL-6. Now, there have been a number of studies shown that IL-6 is in excessive amount in lung, and that leads to cytokine storm and eventually to respiratory failure. Therefore, our approach is to directly deliver this antibody in lungs where it is needed to deplete IL-6. And what we believe is by doing so, we may be able to provide immediate relief to these patients. So that's very critical as to what should be the appropriate administration route. Another thing that I must point out is that if you look at the data from this trial with Roche antibody Actemra that was reported before yesterday, there is also some data which suggests that time to hospital discharge was shorter in patients treated with Actemra. Now, that also implies that patients who were in early stages or moderate stages shows some benefit. And that brings a very important point is that if you are selecting patients who have already who are already requiring oxygen or ventilators, the lungs are already filled with fluid. Therefore, the drug doesn't get access to deeper part of the lung. So our approach is also to go for moderate patients who are not requiring oxygen or ventilators. And our goal is to prevent these patients to hospitalize. And I think that is where our approaches are very distinctive.
Very good, thank you. And our next question, there's some confusion on the ADS ratio. Can you please clarify the change? And additionally, if one were to sell out after the 31st, would they still be entitled to the extra shares? Or if not, when could one sell their shares without losing out on the entitlement?
Yeah, so the stock split of the ADS, first of all, it affects only the ADS in the U.S. Doesn't affect the AIM shares, and what it will do is it will create it's a 1.5 for one, and it will so for every the shares, for example, if the shares were trading at 10. The shares would open up at 4, okay, and you'll have 1.5 times the amount. So as far as the split date is effective as of tomorrow night. So whoever's shareholders as of tomorrow night will on Monday morning have the forward affected split on Monday morning in their account from the 27th to the 31st of July.
And our next question, does the board plan to list Accustem via IPO, or will it be a reverse takeover or merger into an already listed company?
No, so yeah, we're glad that this has been brought up because I know there's been a lot of inaccurate rumors regarding the listing of Accustem. Accustem is planned to go out as a separate IPO onto the standard exchange in the UK. That is the current plan of the board. So there's no. I know there have been a lot of rumors about it going into existing biotech vehicles that are already listed. This is not correct. The plan is for it to be a separate, wholly owned company, not being merged into another at this moment. To just be Accustem. And each shareholder that holds a share of Tiziana Life Sciences as of the record date, which will be established over the next couple of weeks, we'll be entitled to one share for one share of the Tiziana Life Sciences. So it will be a separate IPO vehicle, just Accustem.
Very good. Thank you. And our next question, following the stem printer demerger from the group, are there any plans for any future demergers as we have so many amazing treatments in development? Shadu and I and the board have taken a view that we cannot make a decision on this until we see our data results. Obviously, if our CD3 antibody readout over the next 12 months were positive, that those products are so big that you could have a separate company on that alone, and then we would evaluate at that point in time whether we'd want to spin out into a separate company, Milciclib, which is another interesting product we have, which is quite advanced in the clinic and been in over 300 patients into a different vehicle. So I think until we have more data going into 2021, we cannot commit to doing any more demergers. But it's a great question because we have quite a robust and ample pipeline for small biotech companies, and we believe that the most value is created for shareholders when the story is not too diluted. So when the appropriate time comes, as we did with stem printer, when stem printer was shown to be superior
from an accuracy perspective by between 40 to 45% over Exact Sciences test. We decided that it could be a standalone company, and hence we've pursued the listing of Accustem. When and if we, God willing, have the same type of efficacy data on our CD3 program or potentially our COVID program, then we will look at the future demergence process.
Yes. So if I may add on top of what Gabriel said, so as you know, we've been in touch with several big pharma also. So the possibility of when the data comes out and data are good, so possibility of out-licensing a specific disease indication to one company or another is not completely excluded yet. So our goal is to make sure that shareholders of Tiziana Life Sciences are benefited by doing some business development deal or out-licensing whenever it is possible.
Very good, thank you. And our next question, any update on the start of the trials for Foralumab that we're expected to begin in July?
Yeah, so I can provide update on the Foralumab nasal trial. We've already submitted the package to FDA, and our approach is, and in this COVID-19 situation, what I've been advised is that you submit the package to FDA and FDA comes back with their guidance, and within 30 days after receiving the guidance from FDA, you can initiate the trial, and that is the fastest way. We've already submitted the package for nasal administration of Foralumab in progressive MS, and this trial will be conducted at N. Romney Multiple Sclerosis Center, which is one of the best center for multiple sclerosis. And so that would be starting as soon as we get response from FDA. Regarding the oral administration of Foralumab, that we are in the process of finalizing the clinical protocol, and we would be submitting shortly to FDA again questions and seeking their guidance. This would be a multi-country, multiple sites trial. Total sites could be in between 25 to 30 sites, and it would be in U.S. and Canada, and if need be, in Europe as well.
Very good, thank you. And our next question, is there any way to expedite the COVID treatment due to the ongoing pandemic? You're aiming for Q1 next year, but it could be sooner in light of the urgent need?
Very good question, and I can tell you that we are trying to expedite in every possible way, and we have saved a lot of time. So, for example, GMP manufacturing of anti-IL-6R usually takes anywhere from 14 to 18 months, and we have long-term agreement with Lonza. However, I didn't want to wait for 14 to 18 months for just GMP manufacturing, so we explored other companies, and we reported publicly that we signed agreement with SCC Bio, and they will be able to produce this antibody, GMP antibody, within 8 months. And in parallel, I've also produced non-GMP material, which we are using for inhalation technology development, as well as inhalation toxicology in monkeys. So all these activities are going on in parallel, and we hope to submit IND by February of 2021, and then start the clinical trial. This is the fastest way to do, fastest way to initiate the clinical trial in COVID-19.
Very good, thank you. And our next question, would any of the current therapies possibly be beneficial in the treatment of COPD?
Yeah, very good question. And so I must give you some background on this. So we in-licensed this antibody, anti-IL-6 receptor antibody in 2017. And at the time, our thinking was that we will develop the antibody for treatment of acute respiratory distress syndrome, or ARDS. And so that is what we are still planning to do. However, when we saw the data in March, early March, about Actemra showing positive results in COVID-19, so we initiated program on COVID-19 as well. But I must emphasize here that our antibody is the only antibody which is, number one, fully human, and it is the only antibody that depletes circulating level of IL-6, which is very distinctive feature because circulating level of IL-6 is important for some of the pathologies in ARDS as well as multiple myeloma. So once we have the GMP material available, our plans are, number one, to treat for COVID-19 as well as for other diseases.
Very good. Thank you, Shalu. And that looks like that was the last question from the audience. I guess just in summary, I covered a lot of ground in the presentation as well as here in the Q&A, but just in summary, what's the key takeaway? Why should investors take interest in Tiziana today?
Sure. Well, in summary, the company senior management team has gotten 3 drugs from the discovery phase through the FDA approval phase, which is a very unique characteristic to see in a small biotech company, number one. Number two, we have a very big pipeline. We have Milciclib, which is a phase 2B ready drug. It's been in over 300 clinical patients. We have a phase 2 ready drug for Crohn's disease. We have a phase 2 drug for nasal multiple sclerosis, progressive multiple sclerosis. We have a application indication also with the same nasal CD3 antibody for Alzheimer's disease. We have our COVID program, and we have a stem printer, which is going to be a standalone company and is the best in class if you look at it compared to Oncotype DX's leading product, which is it belongs to Exact Sciences. So if you look at some of the market caps right now in the biotech sector, which has been booming in the last 4 months, and you look at our product pipeline, we believe that there is a big space to grow, especially because the stem printer IPO and valuation could be a potential asset worth a couple of $100 million just by itself.
Yeah, and if I may add to what Gabriel said, what I want to emphasize is our approaches are innovative and quite distinctive. For example, progressive MS or progressive multiple sclerosis. Currently, secondary progressive MS is treated with Ocrevus, which is anti-CD20 antibody. Now, that antibody is publicly known doesn't work very well. And our approach is through activation of immune system through stimulation of T regulatory cells, which are completely depleted in this disease. So that is a totally new approach. And again, the treatment is through nasal administration rather than intravenous. And that is where we believe that our approach is quite distinctive. Same thing goes for Crohn disease. Crohn disease is gut inflammation. Currently, it is treated with subcutaneous. Our approach is to give antibody, which is take-home pill given orally. And that is where what we believe is we would be able to provide topical or local action without immunosuppressing the whole body.
Very good. Very, very exciting. Thank you so much for both for joining us today and sharing the Tiziana story. For everybody in the audience, if you do have further questions after the call, please don't hesitate to reach out to us here at RedChip. You can call us at 1-800-RedChip, or you can email us at info@redchip.com. You can also sign up for free news alerts for Tiziana by going to TLSAinfo.com. That's the U.S., the NASDAQ ticker symbol, TLSAinfo.com. And right at the top left of the screen, you can sign up for free news alerts. There's some interviews, get a copy of the presentation, some additional information there as well. But with that, again, gentlemen, thank you so much for sharing the story. We very much look forward to having you back and sharing some of the successes you continue to achieve some milestones. And again, for our audience, thank you everyone for joining us.
Thank you.
Thank you.